So this is Rick Wychick from NIEHS. So a big project like this that was clearly successful requires a lot of upfront, careful organization. So can you speak just briefly on how was the project conceived, and how did you kick it off, and then how did you decide which player should be involved in this, and who should be doing what, and what type of data infrastructure do you need? Was that all articulated kind of up front, or did you articulate a fluffy, kind of a broad-based vision at first, and then kind of work your way through it as the project matured?
Yeah, so again, I recommend actually reading the Nature commentary I wrote. It touches on quite a few of those points, but not all of them. Just to say, I'm not sure, am I facing you or not facing you?
You are, yes, hi.
Okay.
Hi.
But I think the really important thing is to realize here is that the central NHGRI sort of structured the components of the project, and there was a competitive process inside of these components. So if in that slide where I said data generation, data collection, data coordination center, data analysis center, there were different calls for those. So in some sense, the overall framework was set up by NHGRI. People then proposed, bid for those things, and then NHGRI looked at the resulting winning bids and then adapted a little bit to this. It's worth hearing it from the NHGRI project officer's perspective. I'm not sure if one of them's in there. I often describe this, that scientists are normally like cats, not like dogs. So scientists don't naturally run in a pack. And so asking a bunch of scientists to run in a pack is an interesting process. And frankly, it's better to let the kind of, whenever you can let scientists be cat-like and walk by themselves and do their own thing, it's usually better to just let them be. And so you have to work out where you want to narrow it down and how you want to do that communication. So to try and answer your question concretely, the NHGRI project officers set up the framework, people competitively bid for it, and then there was a to and froing of really working out who is kind of what I describe as the spine of the project. And I must stress here that I am one of perhaps 6 or 8 key people inside of that spine that was very influential in the organization of ENCODE. And if Barbara's there, she can tell you more over coffee, I'm sure.
Thank you.
Hi, Lou Gross, National Institute for Mathematical and Biological Synthesis, and I appreciate the fact that today several staff members from EBI are at our institute working on education for the variety of ensemble and other things. So I appreciate that very much. My question is about your comments about metadata, which I found fascinating. And given your comments about scientists being more like cats, how actually did the iterative process to arrive at an appropriate level of metadata work in terms of the actual details?
Yeah, so I know this is one of those really obvious statements, but you need a good chair of that committee. I mean, you know, no brainer, right? You need someone who will see, understand why the data is useful, and so the key thing to realize here, if you think about it, there's just so much metadata you could capture. I always joke, you know, the phase of the moon. Should we capture the phase of the moon? And as a biologist, you think, well, I'm not sure that's very important. Now, if you're a CERN person in the LHC, it ends up being where the moon is quite important to the LHC experiment. So there's lots of things you could capture about your sample and your experiment and how you did this and stuff like that. And so you need to think forward about the analysis that you're going to do and ask yourself, what do I need to capture? but then you must really see this trade-off about how much the cost of capturing everything. And so my genuine recommendation is to have a small committee, but a committee that spans production and analysis, and have people who really genuinely want to be part of the science. In metadata discussions, quite often you can have people who are very interested in the process of making metadata and in the process of understanding what is done. And that's different from wanting to get the analysis done at the end of the day. So you need a small committee that is really aligned to what you want to get out of this, and you need a good chair.
Thanks.
And they will agonize, and like I said, it doesn't feel comfortable, and that's actually right, but it's not nice. I have to admit.
Thanks.
I'll take the last question. I'm Carolyn Mattingly from NC State University. So do you think that this approach is generalizable? Should it be generalized to other areas of biology, particularly something like we're talking about today with environmental health research?
So I don't think the details are generalizable. I don't think one can take, say, necessarily the structure of encode and map it onto some piece of science in a one-to-one mapping. You really must be driven, like I said at the start, by the science you want to get out. And in some sense, that's the science you want to enable, not necessarily, that's the question. So the question I would pose to you is, what is the science that you want to enable? And then what are the data sets and the information infrastructure that you need to generate to enable that science? But I do think those steps where I said generate, aggregate, organize, transform, analyze, I recognize this in many projects. And so I would be surprised if if some large-scale project doesn't go through those steps. And so I do think there's, of course, commonality at that level. There are some productive things to learn. For example, when I go and talk to colleagues from physics, say astronomy, or I go to the LHC at CERN, I don't learn a great deal about particle physics. It's great fun to go to CERN. But what I do really recognize some similar steps. It's very interesting. And I get some extra insight to my own problem. And so taking a slightly broad view and learning from how other fields have done this is a good thing. And I'm sure, by the way, there are some things that are being done in environmental science at the moment which, biology can learn from as well. So these conversations are never one way. They're always two-way and very useful.
Well, thank you very much for calling in. And did you have anything? Okay. I think we're going to take a short break, and if we could reconvene at 3. Thank you very much, Ewan.